Deep within the human brain lies a special type of cell that might explain how we instantly understand social situations, form deep connections with others, and experience those mysterious gut feelings. They're called spindle neurons, and their story reveals fascinating insights about the biological foundation of intuition, empathy, and social intelligence. Join us as we journey into the microscopic architecture of the brain to discover how these unique cells shape our most human experiences. In 1925, neurologist Konstantin von Economo first described a peculiar type of brain cell that looked nothing like its neighbors. These cells, now called spindle neurons or von Economo neurons, VENs, are found specifically in the anterior cingulate cortex and the fronto-insular cortex of the brain. Unlike typical pyramidal neurons with their triangular cell bodies and multiple dendrites, spindle neurons are large, elongated cells that resemble the spindles used in fabric spinning. These cellular giants are approximately 4 times larger than typical neurons, with cell bodies that can span up to 100 micrometers. What makes them even more special is their streamlined design. They possess fewer dendrites than other neurons, creating a structure optimized for rapid signal transmission between the brain's emotional and cognitive centers. The location of spindle neurons within the brain is no accident. They reside primarily in 2 critical regions: the anterior cingulate cortex, which plays crucial roles in error detection, conflict resolution, and emotional regulation, and the fronto-insular cortex, which helps integrate bodily sensations with emotional states. Together, these regions form key components of what neuroscientists call the brain salience network, the system that determines which stimuli deserve our attention at any given moment. By connecting these regions with their extended axons, spindle neurons create high-speed pathways that integrate emotional, visceral, and cognitive information. This strategic positioning allows them to serve as communication highways between areas processing different aspects of our social and emotional experience, creating a unified perception that we experience as intuitive understanding. The development pattern of spindle neurons tells a fascinating story about their function. Humans are born with very few of these specialized cells, but their numbers increase dramatically during the first 4 years of life. This timeline corresponds precisely with the development of social awareness, emotional intelligence, and theory of mind capabilities in children. As toddlers learn to navigate their social world, recognize others' emotions, and develop empathy, their spindle neuron networks expand and strengthen. This remarkable synchrony between neural development and social cognitive milestones suggests these cells play a fundamental role in our ability to understand others and form meaningful social bonds. The fact that these neurons develop most rapidly during our most socially formative years provides compelling evidence for their role in social cognition. For many years, scientists believed spindle neurons were exclusive to humans and great apes, potentially explaining our unique social capabilities. Then came a surprising discovery in 2006. Researchers found these specialized cells in humpback, fin, killer, and sperm whales. Later studies revealed their presence in elephants and, most surprisingly, certain large-brained birds like corvids, ravens and crows. This distribution creates what scientists call a phylogenetic puzzle. These animals aren't closely related evolutionarily, yet they've all independently evolved the same specialized neuron type. This pattern of convergent evolution provides compelling evidence for the specific function of these cells. Despite their diverse evolutionary origins, species with spindle neurons share remarkable behavioral traits, complex social structures, long-term social bonds, cultural transmission of knowledge, tool use, self-awareness, and apparent empathy. Elephants mourn their dead and comfort distressed herd members. Whales develop distinct artistic traditions and dialects. Corvids craft specialized tools and remember human faces for years. The independent evolution of these neurons in species separated by hundreds of millions of years of evolution underscores how crucial these cells are for navigating complex social landscapes. Researchers now believe spindle neurons serve as the neurobiological basis for what we commonly call intuition or gut feelings. Their anatomical structure allows them to rapidly integrate information across distant brain regions, creating neurological shortcuts that bypass slower, more deliberative processing. When you instantly sense someone is untrustworthy or immediately grasp the emotional atmosphere of a room upon entering, your spindle neurons may be firing. This speed is critical in social situations where milliseconds matter. Regular neurons process information thoroughly but slowly, fine for analytical thinking but too slow for social interactions requiring split-second judgments about others' intentions or emotional states. Spindle neurons appear to create neural shortcuts that allow rapid assessment of complex social situations, enabling what we experience as social intuition. Their streamlined design with fewer dendrites and elongated structure allows signals to travel more efficiently, giving us that immediate sense of understanding without conscious deliberation. The connection between spindle neurons and intuition becomes particularly evident when examining conditions where these cells are damaged. Patients with frontotemporal dementia, which specifically damages areas rich in spindle neurons, display telling symptoms. They lose social awareness, empathy, and the ability to intuitively understand social norms, despite other cognitive functions remaining relatively intact. Similarly, post-mortem studies have found abnormalities in spindle neuron structure and distribution in individuals with autism spectrum disorders, where social intuition is often affected. These clinical observations provide compelling evidence that spindle neurons underpin our ability to read social situations intuitively. When these cells are compromised, people may maintain intellectual abilities while losing the capacity to navigate social waters effortlessly, highlighting the distinction between analytical intelligence and social intuition, and demonstrating the critical role these specialized neurons play in our social functioning. Studying spindle neurons presents unique challenges for neuroscientists. They cannot be investigated in typical laboratory animals like mice or rats, which lack these cells entirely. Ethical considerations limit human brain research to non-invasive imaging techniques or post-mortem tissue examination. Despite these constraints, technological advances are opening new avenues for understanding these enigmatic cells. Recent research using single-cell transcriptomics, a technique that identifies which genes are active in individual cells, has shown that spindle neurons express unique genetic patterns associated with high-speed communication and social cognition. They contain specialized molecular machinery that allows them to maintain their unusual shape and transmit signals over long distances with minimal signal degradation. Some scientists now use brain organoids, miniature lab-grown brain structures, to study how these specialized neurons develop and function, offering unprecedented glimpses into their formation and connectivity. These innovative approaches are helping overcome the inherent difficulties in studying cells that exist primarily in species we cannot easily experiment on. The implications of spindle neuron research extend far beyond fundamental neuroscience. Understanding these cells could revolutionize artificial intelligence approaches to social reasoning and intuitive decision-making, areas where AI currently struggles despite advances in computational power. In medicine, targeting the preservation of these cells could lead to new treatments for conditions affecting social cognition, from autism to frontotemporal dementia. The unique architecture and function of these neurons might inspire new computational models that better mimic human intuitive reasoning, potentially helping bridge the gap between artificial intelligence that excels at data processing but struggles with understanding social contexts and human intentions. As we continue to unravel how these specialized cells develop, function, and sometimes malfunction, we may gain crucial insights that help address some of our most challenging neurological and psychiatric conditions while also advancing our understanding of what makes human cognition unique. The independent evolution of spindle neurons in distantly related species offers a profound insight into the nature of intelligence itself. When animals develop complex social systems where success depends on quickly understanding others' intentions, forming cooperative relationships, and making rapid social judgments, spindle neurons appear to provide a critical neurological advantage. This convergent evolution across mammals and birds separated by hundreds of millions of years of evolution suggests that certain neural solutions may be optimal for specific cognitive challenges. The fact that whales, elephants, great apes, and corvids all independently evolved these specialized cells indicates that the pressures of social living drive specific neurological adaptations. Each of these species lives in complex societies where understanding others, forming alliances, and detecting deception can mean the difference between success and failure. Their shared possession of spindle neurons, despite their vastly different evolutionary histories, suggests these cells represent a neurological solution to the universal challenges of social living. Spindle neurons represent a fascinating example of how specialized cellular architecture underlies some of our most distinctly human capabilities. These cells remind us that what we experience as intuition isn't mystical but neurobiological. A specialized neural system evolved to navigate the most complex environment our ancestors faced, the social world of other minds. In these elongated cells may lie the biological foundation of our social nature, our capacity for empathy, and our ability to understand others with a glance, qualities that define the human experience and link us to other highly social species across the evolutionary tree. When we feel an immediate connection with someone, instantly understand a complex social dynamic, or sense another's emotional state without words, we're experiencing the work of these specialized cells. They form the biological infrastructure that makes possible our deepest connections with others, transforming abstract neural firing patterns into the rich tapestry of human relationships that give meaning to our lives. Spindle neurons represent one of nature's most elegant solutions to the challenge of social living. These specialized cells, with their distinctive architecture and strategic positioning within the brain, provide the neurobiological foundation for our intuitive understanding of others. Their presence across species known for complex social behaviors, from humans and great apes to elephants, whales, and even certain birds, reveals how evolution has repeatedly converged on similar neural solutions to the challenges of social intelligence. As we continue to unravel the mysteries of these remarkable cells, we gain deeper insights into the biological basis of intuition, empathy, and human connection. What we experience as a gut feeling or an instant understanding of a social situation isn't magic, it's the product of specialized neural circuitry evolved specifically for navigating the complex social world. Spindle neurons remind us that even our most ineffable human experiences, our connections with others, our social intuitions, our capacity for empathy, are grounded in biology, in the elegant architecture of cells that evolved to help us understand each other in the blink of an eye. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.